Hello, everybody. My name is Sumitra Mandrekar. I'm very delighted to be part of the International Day for Women in Statistics and Data Science Conference. Over the next few minutes, I hope to give you a perspective, my perspective, on the conduct of clinical trials in oncology over the years. I'm a professor of biostatistics and oncology at the Mayo Clinic. As some of you might be aware, it's been a surge of approvals of agents in oncology. As you can see here, between 2016 to 2021, we've had over 207 FDA approvals for drugs. Either it's subsequent line, add-on therapy, first-line drugs, first-line displacing drugs, and in a number of tumor types, as you can see on this screen here. And also the type of drug approvals have changed over time. Earlier on, you can see this orange thing, those are chemotherapies, which are really systemic therapy where a drug is administered to the entire body system and the hope is that it would target the tumor cells. And then over time, in the early 2000s, we came up with targeted therapy. So they are administered targeting only the tumor cell. And now it's targeting our immune system to boost an immune response against the cancer cells. So even the type of drugs have changed over time. And you can also appreciate that there has been a lot of drugs that are very much within molecularly driven subsets. So there's explosion of new targets, there is explosion of molecularly driven subtypes for cancer treatments. And then the most important part is how does this all impact our endpoints and design? As you can see, this is from the FDA document in 2018, a number of different endpoints. And when can you use these endpoints, either for traditional approval or what FDA coins as accelerated approval? Are they surrogate endpoints? Are they clinical endpoints? What sort of designs to use? And what are the type of endpoints? And also there are new emerging endpoints in oncology, like the minimal residual disease, for leukemia, metastasis-free survival for prostate cancer. So this hopefully gives us a sense of the landscape and how oncology has evolved over the last 2 decades. And a substantial portion of oncology now really focuses on biomarker targets and biomarker-driven trial designs. So I just want to take a couple of minutes to discuss what does it mean, what does the term biomarker actually mean? A biomarker to have importance needs to have clinical validity, which means the result from the biomarker test should actually show an association with a clinical outcome of interest. It should have analytical validity, which means the performance has to be accurate, reliable, and reproducible. So if I move lab to lab, if I did it in my local lab, central lab, then those all results give the same answer. And ultimately, of course, after all these research, what's the utility of these biomarkers for a given patient? So it follows the same basic principles as our guiding principles for any statistical hypothesis and development. So they should follow pre-specification of hypotheses and replication so that you address multiplicity issues. They should have properly randomized studies so you truly understand is the marker predictive or not. And otherwise, what happens is you have a number of biomarker studies that are constantly published in the literature, some of them poorly performed. They overestimate the benefit, and of course, it leads to false inferences. So this is a cartoon I want to start with to kind of say, I know as statisticians, most often we get approached by our collaborators to say, come up with a design, come up with an analysis so that that it actually makes it a significant result. So I see that as, okay, come up with a drug that tells you that you're actually telling the truth when you're probably not. So I think biomarkers are in that space. So we have to be very careful as to how we select biomarkers, how are we testing these biomarkers, how are we validating them, and then moving them into the clinic for routine clinical practice. So this started off, the biomarker-driven trial designs really started off in the early 2000s. So you can see some papers that we worked on, dating back as much as 2009 to, more recently in 2019. So to talk about, where we have moved this needle, it's very simple for us to all know, okay, what does a classical design in any setting, oncology or otherwise, look like, right? We identify patients who are eligible for the trial. We would randomize them to a given treatment or standard of care. And then we would do a number of retrospective analyses based on collected samples. The downside of this is you may not have samples of data available on every randomized patient. So you may end up having a subset that could be biased. So we started off earlier on saying, well, okay, how do we change the needle? We make sure that we actually collect the specimen and assess the marker ahead of time. So biomarker is tested on all patients, but then the treatment and the primary endpoint, is independent of the marker status. But now, because you have the marker and the assessment on all patients, you can plan for retrospective marker subgroup analyses. You can specify hypotheses, pre-specified analyses, and so on. So that's how it started off, with these biomarker incorporated designs. And then came enrichment trial designs, which really meant, you assess the patients for a particular marker status, and then you direct therapy based on the marker evaluation. So if there are positive patients, you can randomize them. If they're negative patients, there is belief that they don't benefit from more drugs. So you either take them off the study because they can just be on observation, or they do just as well on standard of care. So that was, that's the enrichment trial design. So these were the sorts of designs that were, again, lingering when initially biomarkers became the mainstay of oncology trial designs. And then came master protocols. Now, what do master protocols mean? It really means you're trying to bunch a group of questions or studies within a single protocol. So again, there are two basic study designs that come under the master protocols. One is a basket trial design. I will go through here, and then the umbrella trial design. So the basket trial design means you develop a single protocol and you have multiple cohorts. So in cancer, at least historically prior to this, you were always treating patients based on the histology. Like, do you have a lung cancer? Are you squamous cell lung cancer patient? Do you have GI cancer, right? Basket trial design completely changed that paradigm, and it said the whole thing is histology agnostic. So really what matters is that molecular alteration that that patient has. So enroll multiple cohorts defined with a molecular alteration, create a single screening platform and patient identification, and then this is an early, let's see what's happening with this drug in this cohort of patients with this molecular alteration. So these were pretty much single-arm statistical designs to support exploratory or early drug development. So you create a number of cohorts underneath that. Now, if you think of it, philosophically, this sounds, the right thing to do. Even from a clinical standpoint, this probably is the right thing. But if you dig a little deeper on the statistical sides, you can see, okay, what things should I consider when I do basket trial designs? First of all, prognostic heterogeneity, right? Because patients within a given cohort can have different prognosis just because they have a different tumor. Although the belief is that it's, histology agnostic. Is that truly true? Can endpoints like tumor shrinkage perhaps mitigate this issue? So what should be the endpoints in this sort of trial designs? And a lot of these are single arm, so that means we're using historical controls. We all know what the issues are with historical controls. It's a lot more important in this setting because patients are coming from different histology subtypes. So that's also an issue. And finally, multiple testing, right? So everybody says we're going to look at the cohort of patients together, but I know I have enrolled, let's say, 3 GI cancer patients and 2 lung cancer patients, and maybe both lung cancer patients responded. I'm sure they're going to start to want to look at subsets, right? And maybe expand subsets. So I feel like there's going to be a lot of multiple testing issues. And so how is it different than performing several independent single-arm trials? So these are things to consider when a basket trial design is set up. There's, of course, a lot of literature out there that talks about how do you design them appropriately, controlling for these things. But just let's understand the design and some of the issues with the design and how do you address those. And then the second master protocol designs that have come to be more prevalent in oncology are called the umbrella trial design. So think of it like an umbrella, right? So you have a funnel, you have a screening platform, which is a funnel, and then you have, individual studies underneath that funnel. And those individual studies are going to be defined by biomarkers. But these are more late stage study designs. So in other words, you already probably have a good sense of how this drug, the targeted agent, works. in this biomarker specified cohort. Another thing is these are all patients with most often similar histology. So this is the umbrella trial designs are pretty much done within a particular cancer type. So there's already some homogeneity in these, in these patients, except for the biomarker driven alterations. Again, like I said, these are larger phase 1, phase 2, phase 3 sort of studies. And the biggest thing that comes here is how do you assign in all of these, how do you assign patients to these biomarker cohorts, right? Because we all know as a human being, we probably exhibit multiple characteristics. So when you test the tumor for an assay, we probably have, alterations at certain levels, more alterations, maybe some is more pronounced and some is maybe weaker. But then how do you decide, how these patients are assigned? So there's many, again, there's a lot of literature out there. You can prioritize it because some is, based on the prevalence, right? You want to make sure you're filling the low prevalence cohorts. So as soon as you see somebody has that low prevalent marker, you want to assign them, to that cohort. Or you can do a random assignment, or there's other options that people have explored. So I want to talk a little bit about an umbrella screening study that we have within the Alliance for Clinical Trials in Oncology. It's called the Adjuvant Lung Cancer Enrichment Marker Identification and Sequencing Trials. It's 5 integrated trials testing targeted therapy in early lung cancer. We have a big screening trial, where patients submit their tumor tissue, and then we submit additional tissue for investigational genomic analysis. And then we have 4 treatment trials underneath. Of course, 2 of the trials, as I note here, have closed. Data is maturing. We don't have a readout yet. And we have 2 active trials at this point. Interesting part with umbrella protocols, as everybody knows, is you can add and remove arms as you go along. So here we started as screening and 2 trials, but now we are screening and 4 trials with 2 trials closed. Just to talk about the complexity of these trials and how they are set up, this is again early stage lung cancer patients, right? They're all completely resected. Depending on their histology and their molecular testing, they can get funneled into one of these arms. I put an X for the trials that are already closed. The one interesting thing about Alchemist is if a patient can't go on any of these arms, we continue to follow them on the main treatment protocol or the screening protocol so that we can get information on their natural history. We can get information on, okay, are they getting these similar type of drugs off these trials, and what can we learn about those patients and how are they different? And then the other aspect of master protocols that we often don't realize is the complexity of setting up the systems and how all the samples and data flow. So for Alchemist itself, you can see there are a number of systems, right? The site is, of course, the key here where they register the patient, but then they have to enroll the patient in one system. And then there is a specimen tracking system that we have to feed that data in. The clinical data goes into a different system. They send the sample to an external lab. The lab accesses, the clinical database, but the lab also talks to the NCI core resource for biospecimens. So you can again see how complicated this whole setup is, and that any one of these lines, if it breaks down, right, it's going to be a very hard process for us to quickly, get on board and get it fixed. So that's, again, an important aspect when everybody, talks about master protocols. It's exciting because they feel like, oh, it's one less protocol to write. We can bunch all these drugs and all these, regimens into one protocol and it's easy to maintain. It's actually not the case. It takes as much effort or more to maintain these protocols and maintain the systems that back these sorts of complicated studies. So again, Alchemist at a glance, I will tell you, we added 2 new trials, completed accrual for 2 trials. And as we added new trials, different histologies came on board. So we have to figure out how we're going to allocate these new type of patients, but those same patients were screened earlier who didn't have a treatment option at that time, but now they have a treatment option. So what do you do for those patients? How are you going to, deal with IRB approvals? Because if you close down the whole study, all accruing studies are closed. And now that's not very good because, now maybe there's only a portion of the whole master link that you want to close. So how do you do that? We also changed the study design along the way. There were new approval of drugs, so it wasn't ethical to do double-blind placebo-controlled studies. So we had to, unblind the patients, and we had to change the follow-up schedule because now these patients are moved into an observation arm, so data collection changes. And we also had to drop arms on some of those treatment studies because of promising data from other trials. So again, the complexity of maintaining 5 studies in one protocol, should not be minimized. So along the way, like I said, we had to continue to think about coordinating systems. And we also had central lab location transition, right? So it's the same lab, but they moved from New Jersey to the West Coast, and now they are in North Carolina. So there's different people accessing the same specimens and doing the technical reviews of the assays. So how much of that impacts or does not impact, your results? So it's important for you to analyze the trend chronologically. There were things, that just you had more negative results from one lab than the other, who knows? So all of that is important to consider as you keep a master protocol open for a very long time. So I will want to leave with a couple of big ideas when we are thinking of precision medicine trials. Of course, we need the first and foremost thing, of course, is you need a good assay, you need a good target. You want to know what the local and central testing issues are before you embark on it. You need to understand the drugs that you're testing. I think this is often forgotten, right? The first 2 words we pay a lot of attention to, but I think the most important thing to launch and maintain these studies is reliable informatics. If we can't have good integrations between the different databases, if we don't know how to match these patients, and we have to be prepared to be nimble along the way, because there are going to be changes. There will be changes, standard of care changes. There are changes in the eligibility of patients. And so you have to think about how are you going to manage those. And so this is all important because you want to eliminate selection bias, right? Like I said, if you don't have a good matching algorithm, you're probably bringing in bias in how patients are assigned to the different molecularly targeted treatments. All of this means you need a rapid decision making, and something that's less talked about is what do you do when you find incidental findings, right? Because these are all on a research platform. But sometimes these assays, when you run, you end up finding germline mutations. What's the process for communicating it back to the patient and the patient's provider? And what's your role as the research database to do that in a timely manner? And then the logistics, right? The consenting process, again, something that's not well thought out, right? How do you consent a patient when you're getting them into the screening trial? Are you telling them you're providing this? You may or may not go into these studies. You can choose to remain on the screening study. And then when you add new arms, you go back and change all the consenting process, right? You've got to add additional options the patients may have. And sometimes you add additional specimen collection along the way. So now patients that went on the study 3 years ago didn't consent for that. So now, but if you want those specimens, how do you modify the consenting to make sure that you go back and request those patients for samples? Enrollment and tracking, I think I talked quite a bit through, on this. A central genotyping, specimen tracking, handoffs, and again, informatics and data management, I think plays a key role in making these trials a success. So at the end of the day, in my mind, I think of biomarkers as definitely important because, we were here, right? We were just kind of treating everybody the same. Of course, markers have given us a little bit of ability to say, no, all these patients are different from these patients, are different from these patients. So how are they different? Maybe it's the molecular alteration, maybe it is some patient demographics, maybe it's histology. So a marker can be anything. But what is the utility of these biomarkers is you're able to separate this into this. But when you think of the clinical utility, it is really how are you impacting the individual patient? I think that is the key to doing biomarker trial designs. That is the question we as statisticians, biostatisticians collaborating on study designs need to ask. I can design the study in a very statistically clever way, but how is this going to impact the given patient? So that's very important when you're designing these studies and thinking through the different information that I presented, on the other complexities of the design. And I know we have encountered this too, right? Many of us, it's like, yeah, can you give me a sample size that tells me that I can do what I want to do with these number of patients? So I think this is another important piece that we as biostatisticians, I think, carry on our shoulders as to how we are going to be absolute police when it comes to doing the right thing for the patients, making sure it's statistically rigor, but also answering scientific question behind it. So that part of my talk was really on the design and analyses and the current landscape in oncology. And I want to shift gears and give you a flavor for, okay, we do all those trials, we learn about those biomarkers, we know about those targets, but beyond the primary endpoint of these studies, what can we learn from these patient data? So something that we analyzed within the Alliance, again, I'd like to give you a few slides on this, is, okay, we have a lot of patients that withdraw consent, right? Why do they withdraw consent? What impacts consent withdrawal? So we looked at across 12,000 patients from so many of our Alliance trials, and I'd like to present it. And this was presented at ASCO, and we are in the process of submitting it as a manuscript. So recruitment to cancer or any clinical trials always has a lot of attention, right? Everybody wants to know, why is accrual low? Can we accrue, to underserved populations? So that's all important. We talk about accrual enhancement plans, but what we don't always pay attention to is you accrue all these patients, but maybe they don't stay on the study. So if they are not there till to observe the endpoint, or they're not there till, the study is completed, I think, that's a bigger problem. You've got the sample size, but you can't interpret the data. So our goal was to summarize what are the factors, patient and trial, that impact patient withdrawal of consent, and can we identify trends over time? We included all phase 2 and phase 3 trial from the Alliance during the period from 2013 to 2019. We wanted to make sure that all patients had at least 2 years of follow-up, because you want to give everybody an equal chance of being on the study or withdrawing from the study, right? We define patient withdrawal as voluntary termination of informed consent to continue participation in a clinical trial at any point during the conduct of the trial. So they could say they don't want to continue with treatment, they don't want to do anything with your study. Both are withdrawal of consent in my mind. So again, we started with a lot more trials and patients, but many of these, almost like 9 of our trials, didn't use the same form. So as you can see, like I was talking about informatics, right? Things change. We don't collect data the same way. So when we try to look across this large period and so many trials, we don't have data collected the same way. So it's hard for us to interpret what withdrawal of consent actually means. So we only included the trials that all use the same form and the same electronic data capture system. We had a trial that closed very early. We didn't include phase 1 trials. Those are just a different kind of trials. So it's hard to know. And we also had a lot of, like the screening trial I talked about. I mean, that doesn't have a treatment intent. So the goal of this was really looking at treatment trial cohort and understanding what impacts retention in these trials. So here's a quick plot over years on the x-axis. So these are the year of enrollment for these patients, and here's the percentage of withdrawal. So you can kind of see that, yeah, I mean, visually, maybe, yeah, it's gone up. Yeah, we were over, in 2013 trials, we had only about 6% of patients who withdrew consent. And now we are probably close to, 10%. But remember, we didn't get into the pandemic year. So these are all patients that are enrolled, prior to the start of the pandemic. I know that's a question we get asked all the time. And here are other information that we looked at, right? So these are just raw percentages, not adjusted for other factors. So you can see if you have a placebo-controlled randomized study, maybe there's more chances of patient withdrawal. If they did not have radiation, they probably withdrew more. And this was interesting because it was believed that radiation trials are very regimented and they are only given for a short duration of time. So they probably are better at retaining patients, right? This is not like a long follow-up forever. Older patients, maybe a little bit more likely to withdraw consent, and then Hispanic patients, more likely to withdraw consent, right? So now we did a lot of, looking at cross correlations, looking at, univariable analyses and so on. But then our final multivariable model, after some clinical selection and some model selection, really we put, these are the variables we considered in our final model, and you can see age, ethnicity, and trial type and radiation, all came out to be significant. Our initial thought was that probably B, year of enrollment, right, would be significant, but maybe that is, that is no longer important, because as the years roll by, we know that we're doing more complicated trials. We know we are probably enrolling different type of patients. So maybe it's not the year of enrollment, but it is really other trial factors that contribute for patient withdrawal of consent. So really then Hispanic ethnicity, older patient age, and incorporation of placebo predicted for higher patient withdrawal, and radiation trials predicted for patient retention. We're still trying to understand. I mean, in our manuscript, we talk a lot about why some of these might be factors that, that predicted for withdrawal or retention. And so, yeah, I think there's a lot of clinical angle to some of these, why this might be the case. But I guess from a statistics standpoint, it was really important to know that the trends for withdrawal are increasing, and there are factors that can probably predict for why patients withdraw. There's definitely other factors that we have been asked to look at, like, are these surgical trials, are these neoadjuvant studies? I mean, but we don't have a good mix of all type of studies and all type of factors to be able to, do a full analysis. So I think I'm almost towards the end. So really, I think it's important that, trials have become complex. Trials are difficult to run, and we have a hard time recruiting patients. So here is an example of, sometimes patients, wait for a long time before they're eligible for a trial. And we have made it very, very complex, right? We collect so much information, we collect so much data that, patients probably just get tired of being on the study and they just kind of, go out, or even the sites, they feel like there's a lot of turnover. There's so much data we collect. And so I think one of the important things we need to think through is, yeah, it's all great to have good statistics and design, but at the end of the day, when you open a trial, I think, how do you streamline data collection? How do you streamline trials so that you make it easy on the patient, you make it easy on the sites, so that you can actually have informed data collection at the end of the study. So with that, thank you very much for your attention, and I'm very glad to take questions. Thank you very much.